In 2019, a Harvard genetics lab flagged a ghost population, roughly 5% of an ancestry signal that matches no known civilization. That same decade, R1bv88, a haplogroup that mainstream models confine to West Africa, turned up in 9% of Egyptian mummies. Denisovan fragments surfaced in isolated Amazon tribes. Haplogroup X2 appeared on continents separated by oceans. A hidden network written in DNA connected civilizations we were told never made contact. And the moment this evidence emerged, access to it started disappearing. To understand why, you need to know what changed inside one laboratory after 2010. Inside a climate-controlled cleanroom, the sound of air filtration hums over the snap of gloves and the click of pipettes. Since 2010, a single anatomical target has transformed ancient DNA research, the petrous portion of the temporal bone. This dense fragment, hidden at the skull base, can yield over 30% endogenous DNA, sometimes hundreds of times more than long bones or teeth. With this breakthrough, laboratories could finally recover genetic material from remains thousands of years old, even in humid or temperate climates where DNA usually vanishes. But high yield alone is not enough. Ancient DNA is riddled with chemical scars. Cytosine bases deaminate over millennia, turning into uracil and scrambling genetic code. That is where uracil DNA glycosylase, UDG, comes in. By selectively removing most uracil residues, UDG treatment slashes the rate of false C to T mutations, yet preserves a faint signature at the ends of the molecule. This terminal damage pattern is the calling card of authenticity, a molecular timestamp that rules out modern contamination. Every extraction is flanked by negative controls, blank tubes that must stay silent, or the whole batch is tossed. Library preparation happens in a separate facility, with double-indexed barcodes tracing every fragment back to its origin. The lab director says that no one enters the cleanroom without full Tyvek, double gloves, and a UV decontamination cycle. One stray skin cell, and you compromise decades of history. These protocols are not academic exercises. They are the firewall between genuine discovery and self-deception. Only with this rigor can claims of unexpected haplogroups, ghost populations, or cross-continental matches be taken seriously. The technical leap after 2010 did not just open new doors, it set a standard that any extraordinary evidence must now meet. Population genetics has become a numbers game. One played on a scale undreamed of just a decade ago. The backbone of modern ancient DNA analysis is the standardized panel, a set of 1.24 million single nucleotide polymorphisms, SNPs, known as the 1240K panel. This toolkit, now used in nearly every major study, acts like a universal measuring stick. It allows researchers to compare genetic fragments from a Sumerian skull, a Peruvian mummy, or an Indus Valley tooth on equal footing, even when the DNA is patchy or degraded. To qualify, a sample must hit a minimum coverage threshold, often just a few hundred or a few thousand SNPs out of over 1 million. Anything less, and the data are set aside as unreliable. But the real detective work happens with the statistics that sit behind the headlines. D-statistics, sometimes called ABBA-BABA tests, are the workhorses for detecting ancient gene flow. Their formula is D equals ABBA minus BABA, divided by ABBA plus BABA. That comparison looks at patterns of shared alleles across 4 populations. A positive or negative value, if it passes a high Z-score threshold, can point to hidden ancestry or unexpected admixture events. These tests do not just flag a match, they can reveal when a population carries fragments from an unsampled, now vanished group, a so-called ghost lineage. To avoid being fooled by noise, researchers rely on bootstrapping. This means resampling the data hundreds of times to see if the result holds up under pressure. If the signal disappears with a different set of SNPs, the claim does not make it into the published tables. Only robust, repeatable findings survive this statistical gauntlet. Out of these methods comes a new vocabulary: QPADM for modeling mixture proportions, D-statistics for testing gene flow, and ghost lineage modeling for filling gaps in the genetic record. These tools are now the standard for turning raw lab data into claims about ancient migrations, population splits, and the possibility of lost civilizations. The next steps in the investigation will depend on how these numbers are interpreted and who controls the narrative. In 2019, a team at Harvard led by David Reich published findings that unsettled even the most experienced geneticists. Their analysis, using the 1.24 million SNP panel and QPADM mixture models, uncovered a persistent anomaly, a ghost population invisible in the archaeological record, yet contributing around 5% of the ancestry in Bronze Age Europeans and South Asians. No bones, no artifacts, just a genetic signature, surfacing in the data from over 400 ancient genomes, most drawn from petrous bones and rigorously authenticated. The numbers held up under every test, from bootstrapped statistics to d-statistics and F4 ratios. Reich himself described it plainly. We are seeing a signal that should not be there. It is like finding a shadow with no body to cast it. Another surprise emerged from mitochondrial DNA. Haplogroup X2, a rare maternal lineage, appeared in places that made no sense on traditional migration maps. In Montana, the Anzac one child, buried 12,600 years ago and linked to the Clovis culture, carried X2A, a subclade never found in Siberia or East Asia, but present in some Native American groups. Yet X2 also persisted among the Druze in the Levant, the Basques of northern Spain, and scattered ancient remains across Europe. Ancient DNA from the Basque region, sequenced at high coverage, showed X2 diverging from the broader European pool nearly 8,000 years ago, predating the Neolithic spread. In the Levant, X2b survived in isolated mountain communities, resisting later migrations. No accepted model explained how X2 crossed from the Near East to the Americas, skipping Siberia. The standard Beringian migration narrative, in which small bands crossed an Ice Age land bridge, could not account for the patchwork of X2 subclades or their absence in supposed intermediary populations. The evidence, stamped in ancient bone and confirmed by modern sequencing, forced new questions. Was there an earlier migration or a lost maritime route? Or does the ghost's shadow hint at a civilization erased from both memory and record? In the Nile Valley, a quiet revolution began with the analysis of ancient Egyptian mummies. When researchers applied Y chromosome assignment methods to a new data set, something unexpected surfaced. R1BVA8, a lineage usually tied to Western Europe, turned up in 9% of male samples from dynastic Egypt. The numbers came from petrous bone extracts, often less than half a genome's coverage, but with enough authentic reads and S-star statistics to pass contamination checks. The result was not dismissed as a fluke. Multiple labs confirmed the presence, but the pattern did not fit any established migration map. R1BV88 sits on a branch that splits from the main European cluster, suggesting a North African source that predates the classic steppe migrations. The implications unsettled the timeline. If R1b reached the Nile before the Bronze Age, what corridors or forgotten contacts might have carried it there? The surprises did not end in Africa. Far from the Nile, in the humid forests of Amazonia, a different anomaly came into focus. Genome-wide scans of indigenous groups revealed small but consistent Denisovan fragments, about 2% of the genome in some Amazonian populations. These traces should not exist according to the mainstream model. Denisovans, an archaic human branch, are supposed to have left their strongest genetic legacy in Melanesians and certain Asian highlanders, not in South America. Yet using d-statistics and S-star track detection, researchers found archaic segments in Amazonian genomes that resisted every attempt at easy explanation. The fragments did not match Neanderthal or known Eurasian sources. They stood out as a faint, persistent echo of a population long vanished from the fossil record. Each of these findings, R1BV88 in Egypt and Denisovan fragments in the Amazon, passed through the same forensic gauntlet. Negative controls, double-indexed libraries, damage authentication, and statistical bootstrapping. No single contamination event could explain both signals, separated by continents and thousands of years. And yet, neither finding sites comfortably within the accepted story of human migration. The anomalies force a new set of questions. Did ancient mariners cross oceans earlier than believed? Were there lost corridors of contact, erased from both history and the archaeological record? Or are these signals the genetic footprints of a civilization that official narratives have overlooked, or perhaps chosen to ignore? As the roster of anomalies grows, each new sequence intensifies the puzzle. The genetic map of the ancient world is no longer a simple story of gradual diffusion. Instead, it looks more like a network, one that stretches from the Nile to the Amazon and from forgotten tombs to the edges of modern science. Across continents and millennia, megalithic structures rise from the earth in patterns that defy simple explanation. Stonehenge, with its concentric rings and solstice alignments, stands in southern England, while Tiwanaku's massive cut stones loom over the Bolivian Altiplano. The radiocarbon dates 5,500 to 4,000 years ago place their construction within the same broad window, yet mainstream models insist these societies developed in isolation, with no contact or shared blueprint. A closer look at the DNA extracted from burials near these sites reveals subtle clues that challenge this narrative. In 2019, researchers identified a rare single nucleotide polymorphism, SNPQ54, in ancient remains from both the British Isles and the High Andes. The frequency is low, well under 1%, but its presence in both populations, separated by oceans and thousands of kilometers, is difficult to dismiss as coincidence. The 1240K SNP panel, designed to standardize these comparisons, confirms the marker is genuine and not a laboratory artifact. Architectural parallels deepen the mystery. The lintel and post construction at Stonehenge mirrors techniques found at Tiwanaku's Kalasasaya Temple. Both sites are aligned to solstices, both employ massive stones quarried from distant sources, and both cultures left behind little evidence of the tools or methods used to move such weight. These echoes in stone are now joined by faint genetic signals, hinting at either ancient maritime diffusion or a shared inheritance from a forgotten source. If these connections are more than chance, the implications reach far beyond architecture. They suggest a network that once spanned oceans, linking builders not just by knowledge, but by blood. The next question is why such links would vanish from the historical record, and what forces might have erased the memory of a civilization that once circled the globe. Around 12,800 years ago, the planet lurched into a sudden freeze. The Younger Dryas, a climate shock lasting more than 1,000 years, dropped temperatures across the northern hemisphere almost overnight. Ice sheets advanced, rainfall patterns collapsed, and ecosystems unraveled. Archaeologists talk about this period as a break in the story, a bottleneck so severe that entire ways of life disappeared. But the genetic record tells an even sharper tale. In North America, ancient DNA from Clovis-associated burials, like the Anzac I child, shows a population structure that vanishes after the Younger Dryas. Genome-wide data reveal a 15% replacement in the gene pool, a turnover too abrupt for slow migration or gradual drift. It is as if a thread connecting earlier peoples was cut, then rewoven with new material. The same pattern shows up in Eurasia. Mitochondrial lineages and rare Y chromosome markers blink out, only to be replaced by others with no clear local ancestor. Graham Hancock describes the Younger Dryas as the moment the lights went out for an advanced global culture. Randall Carlson points to the impact hypothesis, a comet or asteroid that triggered the cooling and shattered coastal settlements. Whether by cosmic accident or climate alone, the genetic evidence records a sharp contraction, a narrowing of diversity that erased much of what came before. This bottleneck is more than a footnote. It explains why the traces of a possible ancient network, shared haplogroups, megalithic echoes, even rare Denisovan fragments, suddenly grow faint or vanish entirely after 10,800 BCE. The missing links in the DNA may not be hidden, they may have been wiped out, leaving only scattered signals for modern science to recover. For anyone seeking to reconstruct lost civilizations, the Younger Dryas is the event that turned continuity into enigma. The trail of genetic anomalies does not run unopposed. At the heart of the controversy are the institutions tasked with curating the physical and genetic record of humanity. The Smithsonian's accession logs from the early 20th century show a conspicuous absence, hundreds of remains listed as received, then quietly omitted from subsequent inventories. Requests for documentation on these specimens, filed under the Freedom of Information Act, have returned only redacted memos or notices of missing files. In Egypt, the Ministry of Antiquities enforced a strict export ban on all human remains from 1989 to 2017. Even after the embargo lifted, permits for destructive sampling, which is essential for ancient DNA work, were granted only to a handful of government-approved projects, with conditions that barred the removal of bone powder or raw data from the country. Several high-profile genome studies, including one on royal mummies, stalled for years in bureaucratic limbo before a partial dataset was finally released. Barriers extend to the publication process itself. Letters from journal editors preserved in private correspondence archives show repeated requests to omit or downplay ancestry components that diverged from accepted migration models. In at least 2 cases, manuscripts reporting unexpected haplogroups in Egyptian and South American samples were rejected outright, with editors citing a need for additional contextualization, a phrase that in practice meant aligning results with mainstream timelines. Accession numbers for several controversial ancient genomes remain under embargo, sometimes for as long as a decade, preventing outside researchers from reanalyzing the data. In the absence of public raw files, claims of cross-continental links or ghost populations remain trapped behind institutional walls, fueling suspicion that the full story of human origins is being filtered before it reaches the light. 12 Paracas skulls, each with their distinctive elongated shape, became the centerpiece of a genetic controversy that refuses to go away. Brian Forster, an independent researcher, announced that DNA from these skulls showed markers unlike any known Native American group, hinting at possible European or Middle Eastern ancestry. The claim quickly spread across alternative history circles, but the details behind it remain stubbornly unclear. The samples, 12 in total, were said to come from controlled museum collections, yet no formal chain of custody documentation has surfaced. The laboratory report, never published in a peer-reviewed journal, exists only as a summary circulated online and referenced in Forster's own media. Key information is missing. There are no raw sequencing reads, no data deposited in public archives, and no independent replication by outside laboratories. The geneticist involved remains unnamed, and the methodology, down to the extraction kits and reference panels, has never been disclosed. Attempts to replicate the findings have stalled. Certified ancient DNA labs have requested access to the original bone powder or even the extracted DNA, but permission has not been granted. The few technical details that do exist mention low endogenous DNA and ambiguous haplogroup assignments, with no evidence of the rigorous damage authentication or negative controls that have become standard since 2010. Without these checks, the risk of contamination, especially with modern European DNA, cannot be ruled out. Forster himself, when pressed for specifics, has stated only that unknown genetic markers were detected, but the absence of raw data means that no one can confirm or challenge the results. In the world of ancient DNA, extraordinary claims demand extraordinary transparency. The lack of open data and independent verification leaves the Paracas genome story suspended in uncertainty, fueling speculation but offering little substance for science to interrogate. Copper artifacts from Mesopotamia and the Bolivian highlands, separated by half a world, have been subjected to trace element assays. The results show a fingerprint, almost identical ratios of lead, antimony, and silver, despite no known trade route connecting these cultures. Metallurgists and independent labs have confirmed the match, yet the findings sit in technical reports, rarely cited in mainstream syntheses. In the realm of ancient acoustics, the resonance chambers at Malta's Hypogeum, Egypt's Great Pyramid, and Chaco Canyon have all registered peak frequencies near 128 Hz. These measurements, repeated with modern equipment, suggest a shared understanding of sound manipulation, but the official explanations still attribute the effect to chance or unrelated local traditions. The rise of consumer DNA testing has brought the debate into the present. Millions of people now upload their genomes, searching for ancestral roots. Some receive results that hint at unexpected connections, haplogroups, or rare markers with no clear explanation in traditional migration models. Yet the databases that store these genomes are managed by a handful of institutions, and access to ancient samples remains tightly controlled. Requests for raw data from controversial sites are often met with delays or outright refusals, leaving independent researchers to piece together the story from fragments. Meanwhile, the next technological leap is already underway. CRISPR-based techniques promise to recover readable DNA from remains once thought hopelessly degraded, bones exposed to tropical humidity, or charred by ancient fires. The timeline for this breakthrough is measured in years, not decades. If applied to the petrous bones of megalithic builders or the Paracas skulls, these methods could resolve questions that have lingered for a century. The tools exist, the anomalies persist, and the only barrier now is the willingness to apply them openly. The path forward is clear: full transparency, independent replication, and unrestricted access to both data and remains. Only then can the hidden network, if it exists, be mapped in detail and the story of human origins rewritten with evidence that no institution can erase. Today, DNA from Paracas skulls, R1BV88 in Egyptian mummies, and ghost lineages unearthed by Harvard still defy official timelines, remaining locked behind institutional barriers. As new sequencing exposes connections mainstream models call impossible, the question shifts. Is history written in stone or in code? Until buried data sees daylight, the real story of civilization's origins remains just out of reach. What do you believe is still hidden?